Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
We all tend to think of Alzheimer's disease as, something that solely attacks the brain. It steals memories, impacts how we think. But what if that's, only half the story? What if the very proteins linked to this condition are actually causing problems throughout our entire bodies?
That is the really fascinating idea behind some recent research. It's giving us a surprising new perspective on Alzheimer's disease, looking at it as potentially affecting, the whole organism.
The whole organism?
Yeah. By studying the common fruit fly, scientists have discovered that these proteins we know are involved in Alzheimer's. They can have effects that reach far beyond just the brain.
Okay, fruit flies helping us understand Alzheimer's. That sounds intriguing. Tell me about the sources we're diving into today.
Sure. Our main source is a really compelling scientific paper published in the journal Neuron. We're also going to pull in some insights from a related health news article that's from Medical Express, which kind of helps put the science in a broader context.
Right.
And our goal here is really to pull out the most important, the most interesting discoveries for you, give you a clear picture of these widespread effects without, getting totally bogged down in the super technical jargon.
Okay, sounds good. So Alzheimer's, the classic picture, involves these amyloid plaques and tau tangles in the brain. Can you just give us a quick reminder of that traditional understanding?
Absolutely. For a long time, the main focus in understanding Alzheimer's has been on the buildup of a specific protein, amyloid-β42, which forms those plaques, and also the accumulation of another protein, tau, which forms what are called neurofibrillary tangles.
Right, inside the brain cells.
Exactly, within the brain.
Okay.
And these were thought to be the main things driving the damage to nerve cells and, consequently, the cognitive decline we see.
But it sounds like that view is expanding now. We're realizing the disease might have effects beyond just the brain.
Precisely. While the brain is obviously central to Alzheimer's, more recent research has started suggesting that the disease has consequences extending beyond the nervous system. It could potentially affect other organs, other systems in the body.
And this new fruit fly research adds weight to that idea.
It offers strong support for this broader impact, yes.
Okay, but fruit flies, why these tiny insects? How are they useful for studying something as complex as human Alzheimer's?
It seems counterintuitive, maybe. But fruit flies, Drosophila melanogaster is their scientific name, they're actually an incredibly valuable model for studying aging and age-related diseases like Alzheimer's.
Really? Why is that?
Well, despite being, small and seemingly simple, they share a lot of fundamental molecular pathways with mammals, including us humans.
Oh, okay.
So this means that many basic biological processes relevant to diseases like Alzheimer's are often quite similar in flies and people. This lets researchers gain really valuable insights that could potentially apply to human health. Plus, their short lifespans and the fact that their genes are relatively easy to manipulate makes them great for studying how diseases progress over time.
Makes sense. And in this study, they created something called an Alzheimer's disease fly cell atlas. What is that exactly?
Yeah, it's quite a sophisticated tool. Essentially, the researchers built a very detailed map of which genes are active or expressed in individual cells throughout the entire bodies of these fruit flies, both heads and bodies.
Wow, the whole fly.
The whole fly. And these were flies engineered to produce those Alzheimer's-related proteins. This atlas catalogs gene activity in, get this, 219 different cell types.
That's incredibly detailed.
It is. It gave them this really comprehensive view of how these proteins were impacting all sorts of different cells across the entire organism, not just focusing on the brain like usual.
So how do they actually make these Alzheimer's flies?
They used a very precise genetic technique. They made the flies produce either the amyloid-β42 protein or the tau protein, but specifically within their nerve cells, and only once the flies were adults.
Oh, okay. Why only in adults?
That was a really important aspect. It allowed them to study the effects of these proteins during the adult stage of life, avoiding any potential developmental problems that might have confused the results if the proteins were there from the very beginning.
Smart. Okay, so they had these flies making amyloid-β42 in their neurons. What effects did they see?
The main effect of the amyloid-β42 proteins seemed to be primarily on the nervous system, as you might expect. But within the nervous system, it was the sensory nerve cells, the ones responsible for senses like sight, hearing, and smell. They seemed particularly vulnerable.
Huh. That's interesting, especially the smell part. I feel like I've heard that losing your sense of smell can be an early sign in human Alzheimer's.
Exactly. And this study actually identified specific smell-related nerve cells in the flies that were affected by amyloid-beta 42. They even pinpointed certain olfactory receptor neurons, the ones that detect specific smells like vinegar or carbon dioxide, as being the most impacted.
Wow.
And they confirmed this. They showed a significant decrease in both the number of these sensory nerve cell bodies and the signals they were sending out. They also saw fewer auditory sensory neuron nuclei, that's the cell's control center in the flies, making amyloid-beta 42.
So the key thing here seems to be that amyloid-beta 42 isn't just causing general brain trouble. It's specifically hitting sensory systems, which might explain some of those early symptoms in people.
That seems to be the implication, yes. It's quite specific.
Now, you also mentioned something called a lactate dehydrogenase high cluster. What on earth is that?
Right. This is another really interesting find. The researchers identified this group, or cluster, of nerve cells in the amyloid-beta 42 flies that showed unusually high levels of the instructions, the RNA transcript, for making an enzyme called lactate dehydrogenase.
Okay.
What's particularly fascinating is that this cluster didn't seem to be just one type of nerve cell. It looked more like different types of neurons were sort of converging, ending up in a similar state with this high lactate dehydrogenase activity.
Like different paths leading to the same outcome.
Kind of, yeah, in terms of gene activity anyway. They even found more of the actual lactate dehydrogenase protein in different brain regions and even out in the antenna of these flies. But it's important to note the amount of amyloid beta 42 buildup didn't directly track with how high the lactate dehydrogenase was, so it's probably not a simple cause and effect.
So any idea what might be causing this increase in lactate dehydrogenase?
Well, they used a technique called gene ontology analysis. Think of it like looking up gene functions in a dictionary to see what processes are affected. This analysis showed that genes related to something called endoplasmic reticulum stress were really active in this cluster. The endoplasmic reticulum, or ER, is like the cell's protein-making and folding factory. ER stress often means there's a problem like a buildup of misfolded proteins. The machinery is getting jammed.
Oh, okay. Misfolded proteins are a big theme in Alzheimer's.
Exactly. They found several representative genes linked to this stress response were more active. Things like heat shock cognate protein 73, calcium binding protein 1, XOX binding protein 1, several others. They even confirmed more of the heat shock protein itself was present, and it seemed quite widespread in the brain.
And this lactate dehydrogenase and stress response, did it change over time?
Yes. The activity of both lactate dehydrogenase and that heat shock protein gradually increased as the amyloid beta 42 flies got older.
And were some nerve cells affected sooner than others?
It seems so. The auditory sensory neurons, the hearing ones, showed the quickest response to amyloid beta 42, showing those signs of ER stress early on.
And did this stress lead to cell death?
Well, they found evidence pointing that way. There was an increased presence of genes linked to cell death, including one related to apoptosis that's programmed cell death called COLR. This was seen in both the auditory neurons and that lactate dehydrogenase high cluster.
Okay.
And they also saw increased levels of something called cleaved caspase 3, which is a marker scientists use to detect apoptosis in the brains of these flies. Often, it was in the same cells that were showing signs of ER stress.
This is incredibly detailed work for fruit flies. Did they see if any of this, particularly this lactate dehydrogenase cluster, shows up anywhere else, like in mammals or even us?
This is where it gets really, really significant. Yes, they did. They found a similar cluster of nerve cells with high lactate dehydrogenase activity in mouse models of Alzheimer's disease.
Okay, mice too.
And remarkably, even in brain tissue from humans who had Alzheimer's disease.
Wow, so they're conserved across species.
It appears so. In humans, this cluster was made up of multiple types of excitatory neurons, and importantly, it wasn't found in the brains of younger individuals who didn't have Alzheimer's.
That strongly suggests it's a fundamental part of the disease process itself.
It really does. They even identified 233 specific genes that showed increased activity in both the fly and the human lactate dehydrogenase high clusters.
And were these genes related to that ER stress again?
Yes. Among those shared genes were ones related to the unfolded protein response, which is directly linked to ER stress. In the human cluster, they also saw increased activity of genes like nuclear factor erythroid, two like one that's related to one of the fly genes they found, and activating transcription factor 4.
Any other clues from the human cluster?
Yeah, further analysis of the human cluster showed changes in genes involved in mitochondrial function, how cells produce energy. Problems with mitochondria are another known feature of Alzheimer's. And interestingly, they also noted that genes whose protein products are sometimes used as indicators or biomarkers for neurodegeneration in blood or spinal fluid, those genes were also more active in this human cluster.
Okay, so to recap amyloid-beta 42, primary impact seems to be in the nervous system, hitting sensory neurons hard, triggering this stress response cluster involving lactate dehydrogenase and ER stress, and importantly, this cluster seems to exist in mice and humans too.
That's a great summary, yes.
So what about the other main player, the tau protein? What did the study find about its effects? Was it similar?
Actually, no. The findings for tau were quite different. When they made tau protein in the fly neurons, it led to significant changes, but primarily in tissues outside the central nervous system.
Outside, you mean in the rest of the body?
Exactly. In what we call peripheral tissues. These changes included altered fat metabolism, problems with digestion, and reduced fecundity, which basically means lower fertility.
Whoa, okay. That's unexpected. Fat metabolism, digestion, fertility. So it sounds like things that change with, well, just getting older.
You've hit on a key observation. The researchers found that these changes in the tau-producing flies closely resembled changes typically seen with normal aging.
So tau in the brain neurons might be speeding up aging in the rest of the body.
That's what it suggests, yes. That tau expression might accelerate aging processes peripherally. They also saw disruptions in how neurons connect and communicate, factors involved in brain-body communication in these tau flies.
Let's dig into those peripheral changes. What exactly happened with fat metabolism and digestion?
Okay, so in the fat body, which in flies handles functions similar to our fat tissue and liver, they saw this progressive disruption of fat metabolism. Early on, the fat droplets inside the cells got bigger, but later they became smaller and more fragmented.
Interesting.
And they actually found a similar pattern in a mouse model of tau-related disease, a Tauopathy mouse. Regarding digestion, the tau flies showed signs of gut dysplasia, basically, problems in the gut lining. They had fewer mature gut cells, the enterocytes that absorb nutrients, and more intestinal stem cells and their precursors.
Like the gut wasn't renewing properly. And the reduced fertility.
Yes, they specifically saw reduced fertility in the male tau flies. And again, linking back to aging, when they looked closely at the molecular level, they found that various cells, fat body cells, reproductive cells, gut cells, immune-like cells from 20-day-old tau flies looked very much like cells from older 30-day-old control flies.
So the tau flies were biologically older in their peripheral tissues.
It appears so, based on both the number of gene transcripts and the specific gene expression levels. There was a really strong correlation between the changes caused by normal aging and the changes caused by tau expression in these peripheral cell types.
Okay, this is fascinating. How could tau protein being made in the brain's neurons cause all these aging-like changes throughout the body? What's the connection?
That's the million-dollar question, right? The researchers explored this by looking at cell-to-cell communication pathways. They found a large number of altered communication signals in the tau flies, particularly affecting those tissues involved in fat metabolism, digestion, and reproduction. For instance, they saw increased levels of a protein called cadherin N2 in many peripheral cells.
And what about the neurons themselves? Did tau change how they communicate?
Yes. Looking at the tau-expressing neurons, they found changes in genes involved in organizing synapses, those connections between neurons, and also in how nerve fibers grow and how signals are transmitted. They even physically observed an increase in the number of connection points, called synaptic boutons, in the neurons that control the fly's hindgut.
So it could be a combination of altered signals being sent out and changes in the nerve wiring itself that connects the brain to the body.
That seems plausible, yes. It suggests tau is disrupting brain-body communication through multiple avenues.
It really sounds like amyloid-beta 42 and tau, while both linked to Alzheimer's, are doing quite different things to the organism.
Very different things based on this study. Amyloid-beta 42 hits the nervous system hard, especially sensory neurons, activating that conserved stress response. Tau, on the other hand, seems to exert its main influence peripherally, mimicking and maybe accelerating aging and messing with brain-body communication.
Though you mentioned some overlap.
There were some similarities noted, yes. For example, both models showed increased activity of genes related to calmodulin, which is a calcium-binding protein involved in lots of cell processes in their neurons, but the dominant effects were definitely distinct.
Wow. These discoveries, they seem like they could have some pretty significant implications for how we think about Alzheimer's and maybe how we could eventually treat it.
Absolutely. I mean, this really opens
See you
up the
next time.
possibility of finding new ways to detect the disease, maybe new biomarkers, and potentially new therapeutic targets that might exist outside the brain.
Yeah, that's a big shift.
And understanding this complex interplay, this communication between the brain and other organs in neurodegeneration, that feels like a crucial step forward.
And that link between smell decline and specific neurons in the flies. That seems really relevant for early detection, doesn't it?
It really underscores the potential importance of smell loss as an early warning sign, yeah. And the study gives us specific cell types affected by amyloid-β42 to potentially investigate further.
And finding that lactate dehydrogenase cluster in flies, mice, and humans.
That suggests it's a core disease process, right? Maybe something fundamental we could target. And the Tau findings really hammer home that Alzheimer's isn't just a brain disease. The periphery matters.
Plus that whole idea that Tau might accelerate aging elsewhere in the body.
That's just a fascinating and potentially really significant angle for future research, definitely.
Now, we should be fair. This is research using fruit flies, so there must be limitations we need to keep in mind.
Oh, absolutely. That's always critical. Fruit flies, while powerful models, obviously don't perfectly replicate all the complexities of human Alzheimer's disease. That's a given. Also, this study used models where the amyloid-β42 and tau proteins were overexpressed, meaning they were produced at higher levels, probably, than typically occurs naturally in the slow progression of human Alzheimer's.
So it might be an exaggerated effect.
It could be, yeah. And the study mainly looked at RNA levels, the gene instructions, not directly at the protein levels and modifications, which we know are also really important in the disease.
Okay.
And finally, while they found strong correlations and links, the exact mechanisms, the precise step-by-step processes driving these peripheral changes, the communication breakdown, the accelerated aging, those still need more investigation to fully pin down.
Makes sense. Did the researchers themselves comment on the significance of their findings?
They did. One of the lead authors, Dr. Hongji Lee, really emphasized how Alzheimer's is marked by these protein buildups and that creating the fly cell atlas was key to understanding the system-wide effects.
Right, getting that whole body view.
Exactly. Another lead author, Yijin Park, highlighted those distinct impacts amyloid-β42 hitting sensory neurons, tau affecting peripheral tissues.
And the aging link.
Yeah. Dr. Zhu Qiaolu pointed that out, how tau production seemed to mimic aging in peripheral tissues and disrupt that brain-body communication. And Dr. Hugo Bellin, another key author, talked about how this atlas really improves our overall understanding and serves as a valuable resource for other scientists too.
It really does make you think differently about how interconnected our bodies are. So, wrapping this up, what's the main message you think our listeners should take away from this deep dive?
I think it's about considering how seemingly unrelated changes elsewhere in your body just might, be connected to your brain health down the line. This research suggests potential new avenues for early detection and maybe even treatment by looking beyond just the brain.
Broadening the perspective.
Yeah, paying attention to the systemic effects of neurodegenerative diseases and really appreciating the vital role of communication between the brain and all the other organs.
That is definitely a thought-provoking takeaway.
And maybe here's a final thought for you to chew on. If subtle changes, say, in our sense of smell, or maybe shifts in our metabolism, could actually be early clues that something's starting in the brain, how might we need to rethink our whole approach to staying healthy, our overall well-being, especially as we get older?
That definitely leaves you with something to consider. Thank you for taking this fascinating deep dive with me today.
My pleasure.
It's a powerful reminder that digging into the complexities of biology, even in tiny fruit flies, can unlock some really crucial insights into complex human conditions like Alzheimer's.
Thanks for tuning in to the Health Headliner podcast by To-Do Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.